Okay, so welcome back everyone for the next section on AMR threats in agriculture. I'm going to co-chair the section with Andrew Singer from UK Center for Ecology and Hydrology, and I'm Daniel Amwako from the University of Gulf. So we're going to invite our speaker, Ed Cytryn, for this section. Ed is a senior researcher and a director of the Institute of Soil, Water, and Environmental Science at the Volcanic Institute in Israel. His research centers on agricultural dimensions of AMR, focusing on impacts of treated water irrigation, animal husbandry, and aquaculture. He was the coordinator of EU PRIMA, decision-supported based approach for sustainable water reuse application in agriculture production, DSWAP, and has served on numerous committees, panel, and working groups that assess risk in AMR associated with wastewater reuse in antibiotic use. Ed has published 70 manuscripts in leading peer-reviewed journals with an H-index of 42 and an HI index of 70. Please, let's welcome Ed Cytryn. Thank you.
So first, I'd like to really thank the organizers for inviting me. And I'll be speaking a little bit about pathogens and antibiotic resistance in treated wastewater and irrigated crops. And kind of the buzzwords are between ecological barriers and under the radar hazards. And I hope after the talk we'll have some sort of idea of at least the tip of the iceberg of how we understand treated wastewater irrigation and how it's associated with AMR. So coming from Israel, we actually reuse now close to 90% of the treated wastewater. Obviously, being in a region that's semi-arid and arid with a relatively large agricultural sector, there's actually no choice. And as you can see here, this practice was started somewhere in the 1960s, and we've exceeded the 50% mark. So basically, right now, close to 60% of all irrigation water is treated wastewater. And I think it's important, though, to stress that this phenomenon is maybe prevalent in Israel now, but due to global climate change and increasing population, it's becoming more and more prevalent in other regions of the world, in Australia and southeastern United States, and now actually in many, many places in Europe. Now, if we look at microbial factors, and these are criteria for irrigation with treated wastewater in Israel. They're similar to criteria in Australia and California. We can look here and see that for unrestricted irrigation, we're talking about a cutoff of 10 fecal coliforms. In other places, it's E. coli per 100 mLs. And it seems that this standard is relatively okay. We don't see big outbreaks of food-related or irrigated produce-related pathogen outbreaks. But I think it's important to understand that these criteria don't necessarily give us an indication regarding AMR. But this is at least pre-policy starting to change, and we can see this directive of the European, well, it's actually a proposal for the directive of European Parliament and Council concerning wastewater treatment. And we won't go into all the details here, but the bottom line is they're talking about by 2025, wastewater treatment plants that receive over 100,000 person equivalents will have to monitor AMR a number of times per year. And obviously, future wastewater reuse regulations are expected to occur. I mean, if we're already talking about monitoring wastewater treatment plant effluents, it seems to be obvious that this is going to also be expected in produce that's irrigated with treated wastewater. So we're talking about surveillance and regulation. But the big question, and I think a lot of people encounter that question in many aspects, is what do we monitor and what are the cutoffs? And so in my talk, and if we look sort of at the influence here, which can be monitored for wastewater surveillance, and then the effluence, so this is what's happening in much of the Western world. This is what's prevalent in many water-stressed environments. And the question when we look at treated wastewater irrigation is, first of all, what the scope of antibiotic-resistant bacteria and associated ARGs are in the irrigation water. Can we pinpoint hazardous markers? Because as I think we've heard in many lectures here, not all genes are created equal. And these obviously will need to be routinely monitored. The next question is, do these elements persist in soil and crops? And the most difficult, which we see in red, is how can we assess the risks associated with these elements? So we measure a certain gene or a set of genes and bacteria, but how can we really use these to actually make some predictions regarding AMR and public health? So I'm going to touch on a number of studies that have been done in the lab over the years. This is Roberto Marano, who is a PhD student, a very talented PhD student in the lab. And I'll mention that in my lab we're using culture-based and culture-independent methods, and this was a purely qPCR targeted study. As you can see, we analyzed a number of different wastewater treatment plants whose water is used to irrigate various crops. Sometimes the water goes directly to irrigate the crops. In other cases, it's stored in reservoirs. And in addition to the 16S ribosomal RNA gene, which is of course a marker for bacterial abundance, the INT-I marker for class 1 integrons for the integrase gene, QNRS, which is a mobile fluoroquinolone resistance gene, and then we looked at 4 different beta-lactamase genes, TEM, which is common and relatively well established, CTXM, which is associated with ESBL, which is much rarer generally than TEM, and then BLA-AXA and BLA-JES, which we've previously found to be strongly associated with integrons and also strongly linked to wastewater. And I'm just going to briefly summarize in a nutshell all of the results from this study. I will mention that in some cases, we actually had experimental plots where the crops were irrigated in tandem with fresh water in addition to the treated wastewater. I'll also mention that we're talking about a wide variety of irrigation water. Depending on which crops are being irrigated, this can be secondary, low quality, and it can be tertiary, higher quality, and this is something that's important to take into consideration. If we basically take the scope of all these genes, we can see that we have much higher concentrations, obviously, in the effluent than in the freshwater irrigation water. DNQ is detectable but not quantifiable, and ND is not detectable. And so with the exception of INT1 and a few exceptions with BLA-AXA2, we almost don't see any genes in the freshwater. We see a lot in the treated wastewater. And if we try to extrapolate the daily loads of ARGs entering the soil in copies per square meter based on local irrigation processes, we can see a huge amount of genes that are actually going into these soils through irrigation. And this is quite disturbing. However, if we look at gene levels in the soil and also the associated crops, so basically the corn, cabbage, potatoes, etc., represent on the left side the soils of these crops that are being irrigated, on the right side the crops themselves. And what we can see is in most cases the levels are either below detection limits or detectable but not quantifiable. And what was interesting is, and this is one specific example where we tested early season where the water is actually strongly diluted with rainwater, and late season where it's completely treated wastewater. And we don't see a correlation between what's detected in the crop versus what's actually in the water. So obviously, Some of these resistance elements are coming from other sources besides the irrigation water. So combining Roberto's results with a study that we did in 2012, which found that treated wastewater has a negligible impact on antibiotic-resistant bacteria and genes, we came to the conclusion that soil is an ecological barrier that impedes dissemination of resistant bacteria and resistant genes. So despite the very alarming levels of genes that we see in the wastewater, it seems that the ecological process, the ecosystem functioning is actually doing a good job of reducing these levels and perhaps reducing to a point that we cannot see them. But we decided to further test this, and we have a very, very large collection of E. coli that are resistant to cefotaxime. Some of these have been sequenced, so these two, which we'll call for short 32 and 68, actually each have 3 plasmids. One plasmid in each of these bacteria have between 6 and 8, the reds are antibiotic resistance genes, so between 6 and 8 antibiotic resistance genes on one plasmid, so they're highly multi-drug resistant. And if we look at the phenotypes, we see a nice correlation between the genes that they harbor on these plasmids and the resistance profiles of these 2 bacterium. So these are our test bacteria. And what Tamar Kozer was a master's student along with Roberto and Chedi Gupta did were these very, very simple microcosm experiments where we used 2 different types of soils, 2 different concentrations of 68 and 32, the 2 bacteria that I showed before. And we had a very, very easy system where we could use dual antibiotics to specifically fish out these 2 bacteria. So in the controls where we didn't spike the microcosms with the bacteria, we didn't get any growth. And when we did, we were able to get growth. And what we see here, oh, that was a spoiler, sorry. So what we see is that over time, I did it again. Wait a minute. Oh, okay, so now now I can finish my talk now. You already know the end point. But what we see is very nicely how the abundance of these bacteria go down over time in the soil. So I'll mention that we kept these at a set temperature and we irrigated them to keep the moisture level pretty much set. The bacteria only spiked at the beginning, so what you can see is after going into the soil, again, whether it's competition, abiotic stress, or a combination of many factors, they're completely disappearing over time. But then Tamar took the soil samples from these various microcosms and incubated them in LB media overnight, and then we plated using these resistance filters that I talked to before. And with the exception of this one that was not visible after 8 days, we got regrowth in every single one of these, even those that had basically disappeared after 2 weeks. So we basically didn't see growth for 2 weeks, but they're still there. And I think that's a very, very important point because when we say something doesn't exist in a certain environment, it generally means that it's either below detection limit or below limit of quantification. And as we know, bacteria have a capacity to regrow, and the fact that we don't see them doesn't necessarily mean that they're not there. We expanded this now, a very large experiment that Roberto did along with Chedi. Chedi is a very talented bioinformatician. And luckily, we had 3 plots that were irrigated in tandem with either freshwater or treated wastewater. There were 2 orchards, an avocado orchard and almond orchard from the northern part of Israel. And there were these lysometers where we had lettuce, which was irrigated with very, very low quality secondary effluent. So we didn't eat the lettuce, and I wouldn't recommend eating it, but this was sort of a worst case scenario to try to see what's happening. And we used a combination of culture-based and culture-independent methods, both looking directly at what was in the soil and using this enrichment platform that you saw in the microcosms before. And I'm just going to briefly touch on Roberto's results because we have a new set of data, which is, in my opinion, even more exciting that we'll get into. So if we look at the microbial community composition, one thing that's very, very interesting is these are the originals. So we only have 1 point for each water sample in the red, 1 point for each of the 3 soil samples, and these are the enriched samples over here. And what we can see clearly by looking at the microbiome is that when we do these enrichments, we can see this signature of the type of water that was being used for irrigation. It's relatively subtle in the two high-quality orchards that were irrigated with high-quality effluent, but when we look at the very low quality, you can see here these 2 points for freshwater and treated wastewater in the original sample. The communities look quite close to each other, but then now we have triplicates for each. The treated wastewater irrigated in the freshwater, they completely separate. And again, this enrichment is sort of enabling us to see certain things that aren't visible in the direct analyses. And this is, again, looking specifically, and this is based on the metagenomic data, at the families that are differentially abundant in the freshwater versus the treated wastewater. And I won't go into all the details, but I will say that if we look at the enriched freshwater irrigated soil, we see a very, very large proportion of bacillus or Bacilliaceae. And when we look at the treated wastewater enriched, and this is something we don't see in the direct analysis, we see a very large proportion of Enterobacteriaceae. And again, in the orchards, we don't see a difference in the ARGs, but in the lysometers with the low quality irrigation, we do see a significantly higher level of ARGs in the treated wastewater irrigated enriched samples versus the freshwater irrigated enriched samples. So that was pretty cool, and Hagai, who presented a poster yesterday, started his PhD, and we said, well, let's kind of build up on this, and instead of looking at soil, let's specifically look at plants. And again, we had these lettuce lysometers on site at a wastewater treatment plant where there was a pilot module that was treating, but the effluent, again, was secondary effluent that wasn't very high quality. Instead of using LB, based on the literature, BHI is supposed to be sort of indicative of the human gut. So we used BHI, or he did, and under both aerobic and anoxic incubations, since the gut is known to have both oxic and anaerobic areas. And again, this whole cascade of culture-based and culture-independent analyses that we used. And I'll just try to take you through this. What we can see, again, looking at the rhizosphere especially, I didn't mention we looked both at the rhizosphere of the lettuce, so the roots, and the phyllosphere, so the leaves of the lettuce. And a take-home message of what we see here is, again, when we look at the non-enriched, we see a rather diverse community. This is, of course, at the phylum level. If we look at the family level, most of what we identify is under 5%, whereas when we do the enrichment, we see obviously a huge bias. So as a microbial ecologist, we know that this is something that we generally don't want to do. We'll get into that a bit later, but what we do see is that in the freshwater irrigated samples for both the rhizosphere and the phyllosphere, we see almost complete dominance of firmicutes. Whereas we look at the anthropogenic, I forgot to mention that one of these sets of lysometers was amended with chicken manure, so it was a one-time amendment, whereas the other was irrigated with treated wastewater, so irrigated twice daily in tandem with the freshwater irrigation. So again, enrichment of the freshwater, we see a huge dominant of firmicutes, and when we enrich with the when we irrigate with the treated wastewater or the manure, enrichment causes a much higher abundance of Enterobacteriaceae, or in this case, proteobacteria. Again, looking at the differential abundance, so again, looking at the native rhizosphere, and again, these are specific families that are differentially more abundant in specific treatments. So just to make a long story short, we can see that, again, there's a very, very strong impact when we enrich on the level of anthropogenic exposure that we see. So freshwater, we almost don't see any of these. For example, Enterococci, Acinetobacter, Escherichia, and unclassified Enterobacteriaceae, which through the metagenomics we realized were mostly Klebsiella and Enterobacter. And again, we see a much stronger effect in the rhizosphere and the phylosphere, but we do see also an anthropogenic impact in the in the phylosphere as well. And interestingly, when we specifically looked at cefotaxime-resistant E. coli, again, nothing found in the native rhizosphere or the phylosphere. When we do the enrichment, we can see over here, we don't see any of these cefotaxime-resistant Enterobacteriaceae in the freshwater irrigated enrichments, nothing in the manure, but we do see quite a bit in the treated wastewater, and similar but much less prominent is in the phylosphere, and this is something that Qiyi called under the radar. So again, these pathogen indicators, these ARG indicators, aren't evident when we look directly at these samples, but after enrichment, we do see this footprint of the anthropogenic effect, and in this case of the treated wastewater irrigation. Again, through the metagenomic data, we looked at antibiotic resistance genes. So this is sort of an average of the total richness of ARGs, and these are the enriched samples. So we have the enriched freshwater, the enriched chicken litter, and the enriched treated wastewater. So again, you can see a significant increase. These are differentially abundant antibiotic resistance genes from the metagenome. Again, very, very strong signature of these genes in the enriched treated wastewater irrigated samples. And the same was true, and this is from Yuan's group, so even though he's not here, at least there's some data that's being shown. And what's really, really evident here, I think the laser sort of ran out, but what you can see very, very strongly is that there are almost no genes in the enriched freshwater irrigated rhizosphere samples. We see a relatively high number of antibiotic resistance genes in the manure amended enriched samples, and then a huge amount of genes that are detected in the treated wastewater enriched samples. And a similar but less apparent trend is also shown in the phylosphere. And we can say that the rhizosphere is highly deterministic if we look at this, whereas the phylosphere, we see a more stochastic but still anthropogenic under the radar effect relative to the freshwater irrigated enrichments. We then went, and this is Ksenia Rokhina, who's a relatively new PhD student who's doing some very, very interesting results with mating between these isolates that Qiyi has isolated, specifically the cefotaxime-resistant ones, and a model recipient, which we received from Connie Smala, which is resistant to rifampicin and also GFP. So we're basically doing these mating experiments, and what we can see, these are all the recipient strains that have been sequenced. Very, very interestingly is that we see a very, very large scope of horizontal gene transfer frequency between these different strains, and now we're trying to figure out why. But those two with the stars are specifically Qiyi strains. And so you can see that not only did we have enrichment of these cefotaxime-resistant strains, they also have their capacity to transfer plasmids to these recipients. And interestingly, when we look at these, so up here is the original recipient, sorry, the original donor. This is the recipient, the 608, and below, these are whole genome sequencing that were done at UTS of the transconjugant, and what we can see is in addition to CTXM15, we have a number of different ARGs, and we can also see this INC-F1 plasmid. So again, real evidence that this is transferring from one to the other. So just to summarize, it seems, and again, this is maybe a simplistic model, but when we enrich, we see a huge proliferation of bacilli. So again, this is something that is obviously biased, but what we believe is that when we want to do risk assessment, it might be actually beneficial to reduce the number of strains that we have and focus on specific strains. So this is the enrichment effect that we see. When we look at the aerobic versus the anoxic, we see much more Acinetobacter in the aerobic enrichments, Paraclostridium in the anoxic, and then if we specifically look at the anthropogenic effect, we're starting to see Enterococci, Escherichia, Aeromonas, Salmonella, and Enterobacter. We see a profuse group of ARGs, and of course these antibiotic-resistant ESBL, E. coli, and Klebsiella pneumoniae. So to summarize, I'd like to introduce, because it's always nice to say something with an ohm at the end at a conference. So the copiotrophosome is the fraction of fast-growing native and anthropogenically derived bacteria in a specific environment. And it seems that enrichment of roots and leaves in BHI media stimulates copiotrophic bacteria and associated ARGs that are often below limits of detection in direct analyses. More pristine conditions, we see bacillus. And in anthropogenically impacted roots and leaves, the enrichment stimulates pathogen-associated genera like Escherichia, Klebsiella, Acinetobacter, and Aeromonas, including ESBL harboring strains capable of transferring resistance. Future directions. So first of all, we hypothesize that anthropogenically impacted environmental copiotrophosomes are hubs for inter-environmental horizontal gene transfer of ARGs, including to human and animal pathogens. And we've begun to apply enrichment, and I don't know if you've heard Hunter's lecture from last session. So we feel that these enrichment protocols may be very beneficial for microbial risk assessment models in order to really try to understand the risks of irrigation with treated wastewater and obviously many other practices. So with that, thank my lab, the funding agencies, and thank you for listening.
Hi, Nicholas Ashbolt, University of South Australia, CRC SAFE. Thanks for a wonderful coverage there. My question is in relation to the enrichment process you're using. Microbial ecologists don't tend to like enrichments because you're just skewing what was there to something that it wasn't necessarily predominantly there. So my question is, have you looked at trying to use perhaps an older technique where you stimulate ribosomal RNA within cells and use direct detection methodologies, flow cytometry, for example, and labeling, to actually see what was really there, as opposed to what's just enriched?
So we haven't used sophisticated. We obviously, as I mentioned, we used 16S ribosomal RNA targeting and metagenomics of non-enriched samples. I'm a microbial ecologist, and I completely understand. My argument is, though, that a lot of times, especially if we talk about plant-associated soil microbiomes, the complexity is so high that it's very, very hard to make assumptions. And since qPCR and, for example, isolation of cefotaxime-resistant coliforms or E. coli is already sort of in practice, these enrichment methods can be used targeting the specific markers in order to try to make sense. The big question is, and I completely agree with you, is what level does it really matter? So that's something that we're trying to figure out now, and we've started to look at correlations between different quality wastewater and irrigated produce to see if we can see how this interacts.
The second reason why I asked that question is I would have expected to see arcobacters there, and I didn't see any sign of arcobacters in anything you presented. Were they there but just at a low level, or you didn't see them?
So arcobacter we see a lot in direct analysis, for sure.
Yeah.
I only showed the differential abundance, and we focus mostly on those groups that are considered to be pathogen indicators, but we do see arthobacter, and it's definitely there in the non-enriched samples. It seems to go down, so it seems that under these conditions, the firmicutes and the proteobacteria, especially the Enterobacteriaceae, are able to compete better, but yes.
Thank you.
Thank you. Any other question?
Hi, I'm Allison Franklin from US EPA. What was the soil type of the soils that you were working with? The reason I asked is because I'm assuming it might be arid region soils, from where you're located at. And I've asked because I've worked with both arid soils and soils from more temperate climates. And, you know, the more sandy soils, we had to do the enrichment process to even get adequate growth if we're looking at things. But with, you know, a loamy soil or clay loam soil, you don't necessarily have to do that. And if it was an arid soil, have you thought about looking at other types of soils, maybe collaborating with someone? Because it'd be interesting to see if you get the same pattern where you, like a loamy soil, you don't see the ESBL, but then you do an enrichment and you do, or maybe you don't need to do the enrichment with the more loamy clay loam soils.
Yeah, so that's a good question. So the lysometers that I showed in the first Roberto study was a sandy soil. The two orchards were clay loams, I guess. The big problem is, you know, and there we almost didn't see any impact. So it's not clear if the effect is due to the soil or if it's due to the fact that in the lysometers we used a much lower quality water. The challenge is when you use very controlled experiments, they don't necessarily mimic the real life, and in the field experiments it's sometimes hard to find fields that are, you know, with the proper conditions. Now we started doing a much more intensive field study where we'll have the soil type and the conditions as well.
The next talk is going to be Barbara Drigo from Future Industries Institute, University of South Australia.
First of all, I would like to thank the organizer for inviting us to present our work. And I'm Barbara Drigo, as they already mentioned, and today I will talk about enhancing the management of antimicrobial resistance in low and middle income countries, and I will give a focus on Fiji. So the Republic of Fiji is one of the largest economy of South Pacific region and is a relatively high level of a Human Development Index and has a multi-ethnic population of 950 approximately thousand people and has 840,000 visitors per year. Fiji economy is largely agricultural and export a lot of sugar canes, papaya, taro, and kava. More than 68% of the population has access to piped water, and approximately 99% has access to basic sanitation services. Fiji was classified as an upper middle income country by the World Bank in 2019. The life expectancy in Fiji is approximately 68 years in 2020, and the key communicable diseases are tuberculosis, hepatitis, typhoid, and sexually transmitted infection. Key non-communicable diseases are diabetes and coronary health disease and stroke. Diabetic infection has been recognized by the World Health Organization that they are poorly managed in Fijian community, and can be a major AMR incubator. So Fiji is important because it's the first Pacific country to develop and launch a national plan for antimicrobial resistance, and this happened in 2015. The NAP is aligned with the WHO GAP 2015 5 strategic objective, and also in 2015 they established a national IMR committee called NARC, which is recognized in using an interagency approach to combat IMR, and in particular a One Health approach. So we have been working in Fiji under the umbrella of the AMR Fiji project since 2020, and is an ongoing project and is collaborative with several universities in Fiji, including governmental agencies such as the Agricultural Ministry and Environment and Health Ministry. And our aims are to create a baseline information on the presence and type of IMR gene, but as well to identify potential IMR hotspot and provide the basis for identifying IMR risk factor, prevalence, and emerging outbreaks. So we have been applying a One Health approach, and to tackle our aim, we have sampled human isolates, and so in the Colonial War Memorial Hospital, we sampled more than 600 carbapenemase-resistant E. coli, Pseudomonas aeruginosus, and Klebsiella. We also sample animal isolate from different sources: dairy, cow, pig feces, cattle feces, calf feces, and chicken feces, for a total of 400 carbapenemase-resistant E. coli and Pseudomonas aeruginosus and Klebsiella. And we sampled several environmental samples from different matrices, including seawater, sediments, and different rivers with rural, agricultural, and industrial stormwater input, open waste facility, parklands, fountains, wastewater, a lot of wastewater actually. And we also collected metadata and for catchment information and land use, population size, infrastructure, farm size, and practices and geographical details. So we also conducted another experiment in which we sample fresh Qiyi. So Qiyi is or Batissia violacea is a food that is always used in Fiji and often eaten raw. And we use Batissia violacea as a bioindicator for AMR, and we sample this in the major river of Viti Levu Island, which is the most populated island in Fiji. And we sample the Batissia violacea in transect from area which has minimal impact to high impact anthropogenic impact. So what we did culture-dependent method for gram-negative, gram-positive, but we focus in isolating carbapenemase resistance genes, but as well, isolate, cefotaxime-resistant isolate, and colistin-resistance 1. And we did MALDI-TOF and isolate the DNA, and this was a huge task, as you will see shortly, and conducted with several PhD and postdoc from UniSA and several organizations in Fiji. But we also use a One Health genomic toolbox, and we use we quantify biodiversity and community structure using metagenomics, and particularly we focus in bacteria, fungi, viruses and protists, antimicrobial resistant genes, biocide, mobile genetic elements, and virulence-associated genes. Whole genome sequencing was conducted by the university UTS, so Stephen Djordjevic group, and they did phylogenetic tree and genotyping for antimicrobial resistant genes, virulence factor and mobilization. And we use also a qPCR, multiplex qPCR assay for detecting emerging and present human opportunistic pathogen and antimicrobial resistant genes, and we use several databases. So in short, we isolate 7,354 isolate resistant. There were 1,513, and the resistant one, whole genome sequencing, were 904. And here you can see some of the examples of the plates where we isolate. And the first data, we try to understand the variability of the antimicrobial resistant genes and the normalized abundance of them, and try to understand whether there were hotspots or not of antimicrobial resistant genes. And what we find out that the majority of the antimicrobial resistant genes were located in the farm manure, piggery, dairy, and vegetables farm, and in the biosolid, including the open dump and the waste. To a lesser extent in soil, sediment, river water, and marine waters. But the interesting was that the variability was very high in the piggery, dairy, and vegetable farms, and this was explained by the diversity of the antimicrobial resistant genes. And when we look at the diversity of these genes, we found NDM as a resistance, and in particular IMP and VIM, which were not present at that time in the area of the Pacific Islands. So we also looked at the catchment effect on freshwater Qiyi resistome, and we look at the normalized abundance of antimicrobial resistant genes, mobile genetic element, and biocide. And the transects were divided in rural and touristic one, and rural and agriculture, and animal, vegetable, farm, aquaculture, and urbanization. And what we saw was that the rural area in general, they had a different normalized abundance than, of course, the animal, vegetable, farm, and aquaculture and urbanization. So definitely the Qiyi were having the effects, so were getting affected in their resistome and in the biocidal presence and in the mobile genetic presence in their own filtering activity. So when we look at the genus identification and the resistant phenotype that we isolated, the top 5 genus were Stenotrophomonas, Enterobacter, and Pseudomonas providentia, and Acronobacter. And so in particular, Enterobacter and Stenotrophomonas were resistant to carbapenemase, cefotaxime, and colistin, and they were the most abundant species. So we looked further in that, and we look in the Stenotrophomonas one, and this is the amazing work that the postdoc of Stephen Djordjevic group, Ethan, is doing. And I apologize for the figure. You are not meant to read all of this, but I will walk you through. So Stenotrophomonas, we were interested particularly in this bacteria because it's naturally antimicrobial resistant. And there are specific concerns on the ability of Stenotrophomonas to form hospital fomites. So we had Australian 133 Australian isolate and 133 Fijian isolate, which we sample from different type of materials, so soil, sediment, dump, and biosolids. So briefly, what you can see over here, we had several clades, but the most important clade is Stenotrophomonas maltophila sensu strictus, which is the one that is highlighted over there, and I think I don't have the pointer, but in gray. And this particular Stenotrophomonas did have a high abundance of virulence-associated genes and antimicrobial resistant genes, particularly SME and beta-lactamases. Then we look at the Enterobacter, that was our next strain and most abundant one. And in a metagenomic analysis, we noticed that Enterobacter was particularly abundant in the manure. When we look at the Enterococcus and Enterobacter species, we saw that Enterococcus was present in soil, sediment, manure, but not in clean water, while Enterobacter was present again in higher abundance manure. We then look at the isolates, and in the isolates we could find different clade again, and but the most important clade was Enterobacter burgundiensis, which is the blue one over there, which was present in all the materials, and so in water, seawater, manure, and sediment, and was resistant to colistin, cefotaxime, carbapen, and SBLs. Further, we look at the E. coli, and so of course E. coli is very important, and especially in a Fiji setting, is an ubiquitous and enteric bacterium with high level of gene mobility, and so we were looking to see whether it was the primary cause of multi-drug resistant extraintestinal infection, and which are very frequent in Fiji. And we had 424 isolate coming from different sources, particularly the environment, the clinical and agricultural and amino sources in Fiji. And what we could see that there were 2 major pandemic lineage that are ST131, which has a high virulence-associated genes and antimicrobial resistance carriage with specific uropathogenic virulence genes. And then we had the lineage ST410, which was extremely high in IRG carriage, including the NDM, that, if you remember, I did mention at the beginning of my discussion of the results. So the key AMR genes were in total, INTL1 was quite present, but in particular it was IS26, which was in 53.8% of the isolates. So in conclusion, what we found was that phylogenetics reveal close relationship and potential transfer event between multiple sample sites. The representative of the Enterobacteriaceae were the only host of a critical AMR market, IS26, and we had two pandemic lineage, ST131 and ST410. ST410 did carry NDM, and we had a high abundance of IS26 in E. coli key AMR genes. So, and to conclude, I would like to thank all the our funding agencies, so the Australian Centre for International Agricultural Research and Centre for Health and Security and WHO, FIU. Thank you.
Very good presentation. I'm Mohammad Usman Zahir from FAO's regional office for Asia and Pacific in Bangkok. So I just had to have two questions and one invitation for you. First question is, you have data from the agri-food sectors as well in your study that you have presented. I wonder if these data are also shared with the relevant agriculture or livestock ministry in Fiji. And the second part is, I wonder, you mentioned, I think FAO, if I may have missed it, but were you working with the FAO's sub-regional office for the Pacific Islands in Fiji, Suva, or it was the regional office? And the third one is just an invitation to you that we are doing a workshop tomorrow, the workshop number 1, where we will be discussing about developing guidelines for Asia-Pacific region for AMR in the animal farm environment. And I believe the work you have done, you could bring a great value to the workshop tomorrow. So if you have not already registered, feel free to register, and if you are free. Thank you.
Thank you. So to answer to your question, the first one, yes, we work with the Agriculture Ministry, and Eric Rafai is our sponsor this project. And also we work with the Ministry of Health of Fiji. And yes, we share our data with them. So actually they have all the collection of our isolate. And thank you for the invitation for the workshop too. I will be there. And yeah, I think I addressed all. Yeah.
So, very impressive study. Congratulations. This is really a curiosity question. So we know that in E. coli we have very many plasmids typically, also carrying these IS elements and transposons and ARCs. But what about Stenotrophomonas and Enterobacter? How abundant are plasmids in these genera?
So for that, Cornelia, you will have to wait the paper that is coming out very soon. I think Stephen and so yeah, we are also looking at the plasmid, and there was no time to present everything over here. But Ethan is working on plasmid and mobile genetic elements in the Stenotrophomonas, and the paper should be out very soon. So yeah.
All right. Thanks, Barbara. Sort of a philosophical question. If, when you look at the strains, if you can sort of make conclusions regarding the impact of those strains you see in the hospital, so the human impact on the environment versus the environmental impact that is coming back to the hospital.
Yeah, so in the phylogenetic analysis, you see we combine, and I didn't mention everything because there is a lot of data over there. So there are the isolate coming from the hospital combined with the isolate coming from the environment, and some of them, they share the lineage. But for instance, the ST410, if I recall it correctly, is mainly present in Fiji, right, Stephen? And so, we also are trying to finalize the paper very soon, so I don't want to spoil everything right now, because, and I would like that Ethan reply also to that. Yeah.
Our next talk is Karina Jin, who is from the National University of Singapore. Welcome.
Okay, good afternoon, everyone. Thanks very much to the organizers for allowing me this opportunity to share with you some of the work that we're doing in Singapore. What I will be talking about today actually is not quite, when I saw the title of this session, I thought, oh, I wonder whether they put me in the wrong session. But what I would like to do is share with you some of the work that we've been doing. Like most of you, we've been collecting a lot of data in the past few years, and at the end of the day, really we're asking ourselves, well, how significant is the data that we're collecting, and does it make sense for the agencies when we collect it? What does it mean to them? So this is where the risk assessment models would be very helpful to help them to interpret the data, and this is what we've been trying to do in, more recently. So what I will share with you is some work that we've been doing in the area of model development, especially with this relative risk model we've recently looked at, where we try to put together data on antibiotic-resistant bacteria, the ARBs, as well as the ARGs. This model can either combine the two together or look at them independently. And with that, we hope to be able to assess, compare the risks, and look at different regions, different seasons, and at the end of the day, give our agencies a way to assess the relative risks. I actually have two other models, but I will talk about them tomorrow in our workshop. If you come, that will be workshop 9. So I'll try to, encourage you to come to that. So this relative risk model is basically trying to give a weighted risk assessment from both the ARBs as well as the ARGs for water samples from diverse environmental sources. What we look at is computing a risk score, which, as you know, risk would depend on the likelihood times the severity of the ARB or the ARG. For likelihood, we make use of the occurrence data, so that would be the actual concentration or abundance data of the ARG or ARB. And for the severity, we make use of these risk coefficients to assess how, or what is the health what is the health burden or the significance of the infection. At the end of the day, we combine this and then we normalize the data so that we can have a comparison across different locations and different seasons, etc., so that we can identify hotspots. So that's our intention for this particular model. So as you know, risk means different things to different people, and depending on what data you have, you can assess risk differently. But this is quite specific. We're trying to identify hotspots with this relative risk model. So on the left-hand side is a sort of simple schematic of what the model looks like. I'll just walk through it very briefly with you, just to show you the framework. So we can use either ARB or ARG, or both, as inputs into the model. And the first thing we need to do is put it on the same relative platform. So we scale the data using minimum, maximum normalization. From there, we have the occurrence data. We then need to look at the severity of the ARB and ARG. So here we use risk coefficients based on literature data. So for ARB, we extract the risk information from Cassini et al.'s paper, which I will expand later in a moment. And for the ARGs, we extracted the risks from the ARG ranker database. Next, we multiply the scaled value from the min-max normalization by the risk coefficient to get the risk score for each individual ARB and ARG. And then we sum up the risk scores and normalize it, and then we have basically a risk score for ARB and a risk score for ARG. Now, this is where it gets a bit subjective, because we then assign a weight to to both the ARB and the ARG, depending on how we feel, how significant is the relative importance of the ARB and ARG. So for our purposes, we used a value of 0.7, but of course, others may choose something different. We did do a statistical analysis, and basically between about 0.6 to 0.8, it didn't really affect the results very significantly. So maybe to expand a little bit more about how the risk coefficients were obtained, right? So these tables show the risk coefficients on the left for the ARB. So this was taken from the WHO DALI's database, looking at the prioritized bacteria, antibiotic-resistant bacteria. So you can see there's six. There's 6 of them listed, and from the DALI database, we would have a burden coefficient assigned, and we can then sort of assign priority ranks to these different bacteria. So, for example, priority rank 1 would have the highest burden coefficient of 37.2, and then the rest is relative to that. On the right-hand table, we have the risk coefficients from the ARG ranker database. So the priority ranks given in that database are listed as such, but you'll see that the risk rank which we actually assign to the ARG is the opposite. So the highest priority rank, say BLA KPC has a priority rank of 1, but we assign a risk rank of 5 to give it a higher weightage as such. So we applied this model, this relative risk model, to data that we have collected over the years in Singapore. And Singapore, as you might know, is a very highly densely populated country. We've got like 5.6 million people packed onto 730 squared kilometers. So with this very high density population, we are in very close proximity to our water bodies. And so, the question of protecting our water resources is critical for us, and we want to also try to minimize this AMR issue. So we have collected various water samples across different environments over the years. We've looked at catchments and reservoirs. We've looked at our seawater, aquaculture, agriculture, hospital wastewaters, and domestic wastewaters from healthy communities as well as foreign communities. We've looked at pets, wildlife, and also our drinking water factories, new water factory, which uses reclaimed water, as well as our typical wastewater treatment plant. So for all these samples, we've collected data on antibiotic-resistant genes, resistant bacteria, as well as antimicrobials. But of course, because it's done over like many years, the methods have evolved over time, and that's part of the difficulty also in trying to compare across the years, because the methods change slightly as well. Okay, so we applied this relative risk model, first of all, to our catchment and reservoir data, which allowed us to take the ARB as well as the ARG. We ran it through this relative risk model and then used it to assess the risk for the different land uses as well as different seasons. So on the right-hand side, you'll see the weighted AMR risk score for the different land use types, which covered urbanized areas, residential areas, commercialized areas, agriculture, and parkland. And on the left-hand graph, you'll see the seasonal comparison of the risk scores. So from this example, you can see that the highest risk score was for the December samples, which really coincided with our northeast monsoon. So it's a very rainy period and a lot of storm flushing from our catchments, and that resulted in the higher AMR risk score. On the right-hand side, you see the risk score for the different land use, different catchments, and here agricultural land, the one in blue, shows the higher risk score. So this is one of the applications of our relative risk model, which allows us to identify the hotspots, both in time as well as in space. Next, we applied the data across a more diverse set of samples. So here we just used ARGs. We didn't have the ARBs. And you'll see that on the left-hand side, we have reservoir and catchments. Catchments had a higher risk score. And then we had aquaculture, which showed higher risk compared to the beaches and the fish that we sampled. And then on the right-hand side, you'll see different wastewater types, including community wastewaters. Now, the far right panel, which unfortunately I can't really point to, the wastewater influence had the highest risk scores. And, but after treatment, you'll see that the risk scores went right down. But you'll notice that there's one really high one, and that's actually the sludge that came from the wastewater treatment plant that had the highest risk. And then we had membrane bioreacted wastewater, treated effluent, whoops. So that one showed the least risk score, which was just consistent with what we expected. So on the right-hand side is a heat map, and on the heat map, again, unfortunately, I can't point, but we see that BLA SHV and BLA, I think it was CTX, are the dominant ARGs, and again, we see that the wastewater sludge actually contains the most abundant ARGs. So I'm running out of time, so I'll just skip to this part. We also collected metagenomes for these samples, and we used the ARG ranker and MetaCompare2 methods to also obtain the risks. And I think since this has been covered in other presentations, I won't go through it, but just to show you the results. So in this case, we compared the ARG ranker, where we looked at the rank 1 and the rank 2 risks for our water samples. And generally, most of the, as expected, most of the risks come from the wastewater samples, especially for the rank 2, which is on the right-hand panel. And then on this one, we use MetaCompare2 to show the risks. Now, for the ARG ranker, the data that we got here, the results were consistent with our relative risk model. For the MetaCompare2, the human health risk score compared favorably with our relative risk model, but we did have some differences with the ecological risk assessment, which is on the left-hand side. And I think I just want to point out to you on the left-hand graph, the last two bars, one shows the MBR, which had the highest or very high risk score from MetaCompare2, which was somewhat inconsistent with what we had from the ARG ranker model as well as our own relative risk model. And the other thing that was different was that the sludge was much lower compared to what we had previously. So with that, just to show you what we have in terms of the relative risk model, that we can use ARB and ARG data together to get some assessment of the hotspots in our environmental samples. Okay, so thank you, and if you have any questions, please feel free to ask.
Zahir again from FAO's regional office in Asia and the Pacific. Very interesting presentation. I just have not the questions about your methods or whatever, but my question is, how do you plan to share these data and the findings that you have from your study with the agencies in Singapore, like your newly established One Health Office or National Parks Board or Singapore Food Agency, or the Singapore generally publishes an annual report on AMR. So how do you plan that these findings get into that annual report that comes out of the federal agencies in Singapore so that it gets to the different stakeholders in the country?
Absolutely,
So thank you.
So actually, most of our funding comes from these agencies, so we directly report our results to them. What they want to do with it is up to them, but currently we have funding from our Singapore Food Agency, our Public Utilities Board, and our National Environment Agency. So at least 3 of these are active funders for our projects. And we've given them the data, and we've worked on different models, and so we've passed it to them. And we're now in the process of trying to consolidate the models and also try to explain and work also with our partners here, with Amy Pruden and Kerry Hamilton. We hope to, try to share more of these models and risk assessments with them moving forward. Yes.
One last question.
Thank you. Very nice presentation. I just wanted to ask you if you have come across the new risk ranks, the 4 different risk ranks by a publication in Nature, and how they are related to the current risk scores that you have in terms of the criteria they used for its association with humans, gene mobility, and escape for those 4 risks rank 1, 2, 3, and 4.
Oh, so you mean the ARG ranker?
Yes.
Yes, so in fact, actually, that's the one that we base our ARG relative risk model on, but
Okay.
we focus on the risk 1 and risk 2
Okay.
because the priority for our agencies was more on the human health related. So it's on the escape pathogens and mobile genetic elements.
I don't know. So do you know if anyone keeps a database for it? Because they had a very tall list of unassessed genes relating to where to relate it with respect to the ranks to put in.
Sorry.
I'm saying that I don't know whether you know anybody keeps a database of it, because they had a high number of unassessed genes with respect to which ranks to put in.
Oh, well, I know that our hospitals, they do have, I think they do keep some kind of a database already, but like this One Health approach right now, I think the agencies are really trying to go to the different sectors, the animal health sector and the environment, and we're now trying to put the whole thing together. But the hospitals are the ones which actually have the main database.
Our next speaker is Rahat Zahir, who is from the Agriculture and Agri-Food Canada.
Right. Thank you so much for the opportunity to present my work at EDAR. I'll discuss some of the research that we have been doing to understand the antimicrobial resistance and its association with mobile genetic elements, and specifically the integrative and conjugative elements. So bovine respiratory disease is most economically significant health issue in beef cattle production system, and it is also known as shipping fever, and that is influenced by stress of moving animals, the immune status those animals have, and the interactions of various bacterial as well as viral pathogens. So the main bacterial pathogens include Mannheimia hemolytica, Pasteurella maltosida, and Histophilus somni, all from the Pasteurellacea family, and also molecute, which is the Mycoplasma bovis, in addition to some viral pathogens. Sorry, I should have moved my slide. So there are a bunch of viral pathogens, so it's basically a very complex pathogenic community. So the today, most of my presentation is about Pasteurellacea bacteria, the antimicrobial resistant genes, or ARGs they harbor, localization of these ARGs in the genome, and the potential of horizontal AMR transfer in these bacteria. In the livestock, animal antimicrobials are used for therapeutic purposes, for the treatment of animals with infection, as well as for the prophylaxis during the period of high disease susceptibility, and also as a metaphylaxis, where there is a disease outbreak in the herd, and the rest of the not yet infected animals need to be protected. So basically, among mobile genetic elements, including plasmids, phages, transposons, and all kinds of integrative and conjugative elements, or ICE in short, ICE could be one of the among very crucial players in the spread of multi-drug resistance, as they often contain the multiple ARGs and cassettes with the some associated insertion elements as well. And in a single genetic transfer event, they can generate multi-drug resistant bacteria or sometimes extreme drug resistant bacteria. So the life cycle of ICE is kind of, they are self-transmissible. They are integrated into the host, but they code the regulatory system to control their excision from the chromosome and their conjugative transfer between the organisms. So basically, this makes them elegant but very, very scary. And the transfer generally happens with through the type 4 secretion system. So we have in the past identified ice in the BRD pathogens from beef cattle, and that contain resistance for up to 7 different classes of antimicrobials with a lot of genes. I think it was like 12 or 13 genes in there. And we also have shown, we also are, in the next few slides, I will show that they also exhibits the horizontal gene transfer potential, and I'll show you in the next slides, a few slides. So these are the, sorry, I'm always late clicking. The antimicrobial resistant genes which were present in those eyes. So we have been collecting deep nasopharyngeal swab samples from beef cattle feedlots in the past several years within various projects, and the swabs are processed for bacterial isolation. Isolates are tested for AMR phenotype, and the whole genome sequencing is done on subset of isolates. So we do just triaging of isolates for. And here you can see that we had sequenced a total of recently 475 isolates, which were sequenced, and out of those, I think 273 had ARGs. Those were present associated with ice. The linkage of ARGs with 3 BRD bacterial species is shown in this cord diagram. And the ARGs, you can see that ARGs including TET-H, SUL2, and bunch of aminoglycoside resistance genes were more prevalent, and those were present in pretty much all 3 pathogens, whereas some other genes were less common, but still prevalent, I would say, for macrolide resistant gene, MSRE-MPHE. MSRE-MPHE is basically one operon, so wherever you find one gene, you find the other one. The ERM-F and ERM42 was also quite abundant. This is another way of looking at the same. Basically, so among ice across 3 species, we found 43, or I think 44, different combinations of ARG-carrying ice. And some of those isolates carried up to 15 ARGs. And we also found that in some cases, for macrolides and for tetracycline-resistant genes, there was 2 copies, one on the ice, and then there was another plasmid, which also carried another copy. So it was kind of additive effect, basically. So in this figure, you can see that some of those profiles are very common across the species, while other genes are more specifically present in certain bacterial species. I think ERM-F and DFRA, probably those were associated with Histophilus. I can't point. Yeah, it doesn't work. The colors on the left-hand side indicate the species, and on the right-hand, on the top, you see the antimicrobial resistance classes to which those resistant genes belong to. And in all 3 species, we found out that the ice was integrated at the leucine transfer RNA site. The whole genome sequence data from our lab also shows that, so this is a little bit high resolution when we are comparing the regions, shows that ice in Pasteuracea have a lot of similarities, not only genes, but genes are 100% identical. And this figure shows the alignment of ice regions from a small subset of Pasteurella multacida, Mannheimia hematitica, and Histophilus somni. And this is a kind of an evidence as well of horizontal gene transfer that seems to be happening across the BRD bacteria. So we established the bacterial conjugation assays in the lab to investigate the horizontal gene transfer of ice within and across species of BRD pathogens, as well as to the pathogens of human health importance. And drift-resistant strains were generated in otherwise susceptible potential recipient strains from various species. So in the next slide, I'll show you 2 examples of the ice transfer from donor to recipient. So this slide shows the schematic integration of ice after conjugation. The transconjugants were subjected to whole genome sequencing. Isolates were confirmed to have full transfer of ice, and we were able to identify the integration sites in the host genomes. So in this figure, the genomic context of ice in a successful conjugation recipients from Mannerheimia is shown. We also observed that in some of the recipient species, the ice sequences did not integrate in the chromosome, but were present as a circular molecule, and the isolate did show the AMR phenotype of the transfer genes. What is the stability of that circular ice? It needs to be tested through subsequent cultures without any selection pressure. So we have basically established in our lab that ice-carrying BRD pathogens can transfer ice to not only within and across their own species, but also to other pathogens of human importance, so including E. coli, Haemophilus species, and Klebsiella and Salmonella. We have been also collecting indicator bacteria, including Enterococcus and E. coli from livestock productions, feedlots, and across the One Health sectors, and we have published quite a bit of that work already. This slide shows an aerial view of a typical Western Canadian commercial feedlot, and this type of a feedlot can host up to 15,000 to 30,000 heads of cattle, and on the side you see the drainage basin. So basically we collect samples from feces as well as from the catchment basin. So we have previously shown that Enterococcus hierae species is predominantly found up to 90% in cattle, while the human opportunistic pathogens, which are mostly Enterococcus faecalis and faecium, are less prevalent in cattle. So among Enterococcus species isolated from feedlots, we have found, so we used Enterococcus hierae, which were predominant, and also faecalis and faecium, and all from bovine feces as well as from the catchment basins. And we have found that a lot of ARGs were present on the chromosome with signatures of ice, transposable elements, and sometimes viruses as well. So we are parsing that data out right now to see, what they belong to exactly, and if those are transmissible through conjugation or not. And we also see plasmids, but so there is less diversity of ARG profiles in Enterococcus, of course, than in the BRD pathogens. And so here you can see, pointer is not working, that you have the Enterococcus hierae, where predominantly you have tetracycline and macrolide RMB, and it's basically a reflection of the antimicrobial use. These 2 antimicrobials are used in Canadian feedlots all the time. So in conclusion, this study shows that the widespread that the ARG-associated ice are widespread, and in the Pasteurelli family, and that there is a link, there is a risk of multi-drug resistant spread followed by horizontal gene transfer from these bacteria. And also, compared to Pasteurelli ice, Enterococcus ice from bovine sources appear to be of low risk. And I would like to acknowledge the research team at AFC and various collaborators and funding agencies, and thank you so much for listening, and I'd be happy to answer questions.
Yes, very interesting. And my question is about your ice elements on the plasmid part. So the ice element have their ARGs and so on. You are interested in those, but they are also composed of phage parts and plasmid parts. Typically, ice elements have this, and I think it's the same here. What type of plasmid is the backbone for your ice elements?
It's a very interesting question, and we need to dig deeper when it comes to the ice. But it seems to be that those cassettes are sometimes present on certain plasmids as well, right? So you would find the insertion elements and bunch of transposases. So the ice we are finding can be up to, in Pasteuracea family, but the ice we are finding are up to 90 kilobases in long, basically. So it seems like they have been collecting those genes from wherever, and then they are now really, really stable and diverse, basically. So it will be really hard to tease apart, I would say, that what genes are originating from. That's your question, right? What genes are originating, ARGs are originating from phages, bacteriophages, and what are originating from the...
Yeah, so the conservative part of ice elements, they are
Yes.
typically composed
Yes.
of plasmid part and a phage part.
Right.
And my question was mainly, what type of plasmids do you have there? I guess based on the sequences you can find out.
We have been seeing a mixture. We have been seeing a mixture of those. And some of those ice-carrying genes, ice-associated genes, the integrases and the those are always conserved, regardless, what's in between.
So welcome everyone. So I'll start. So as you know, farms are reservoir and hotspot of ARB and ARG, but also high and important producers of bioaerosols, which are heterogeneous biological particles suspended in the air that can contain ARB and ARG. And in the farm, their sources are feces, animal litter, and feed. And once aerosolized in the air-down air of farms, some will stay airborne for various amount of time, but others will be dragged outdoor by the ventilation system via the fan exhaust. And so just an example why bioaerosols in farms is really important to study is if we have, for example, the ventilation rate, which is the number of cubic meters of air that is expelled outside the barn via the fan exhaust per hour, and that we know, for example, the number of bacteria indoor the farm per cubic meters of air, and that we multiply those factors, we'll have emission rates, which are number of bacterial genome expelled from the buildings per hour, and this is the real data from my experiments. So about 10 to 11 airborne bacteria will be emitted from farms and will be expelled outdoor when they can disseminate and participate, if they contain ARB and ARGs, to the dissemination and risk of AMR transmission. So the objective of what I'm going to present to you today is part of my PhD project, where we really want to characterize the diversity of ARGs and bacteria emitted from pig finishing buildings and broiler chicken barns and in their vicinity. And in Canada, Quebec is the first pork producer and the second poultry producer. This is why we focused on research on those two production types. And then just a quick overview of the 4 farms that we sampled, so 2 pig finishing buildings and 2 broiler chicken barns. Pigs were fed tetracycline in their feed, so we administered tetracycline in their feed, while broilers it was bacitracin and 2 anticoccidial drugs. You can also see the number of animals in the buildings, as well as their weight throughout the sampling period, as well as different animal lots while we were doing the sampling, and the number of buildings on sites in the production types. And for the air sampling, we use filter-based air samplers, so air is collected and trapped on filters. We have 2 types of air filters, the SAS 3100 that will collect 20 cubic meters of air per hour, and the bigger one, the SAS 4100, which collect 264 cubic meters of air per hour. We also record the temperature, relative humidity, wind direction, and wind speed. And we visit each farm 3 times in the warm season, which means maximum ventilation, so maximum emission outside the buildings of bioaerosols. And then for the sampling points, we have 6 in total. So 1 indoor, the farm, 1 directly at the fan exhaust, and after, depending on the prevailing wind direction, 10 meters, 100 meters, 1 kilometer downwind the farm, and 1 kilometer upwind the farm. And then while we have our SAS filters, we're going to elute the viral source trap in the filters, extract DNA. We will quantify the total bacteria with the 16S RNA gene. So we will have gene copies of 16S per cubic meters of air. And then after, we want to detect some ARGs, so we use the high-throughput PCR platform, the Takara Bio1, with a panel of 27 ARGs and 3 mobile genetic elements that are listed here with different antibiotic families. And we use the 16S as the reference for normalization to have ARG copies per cubic meters of air. And finally, we did bacterial diversity with amplicon-based sequencing. So here for the results, it's the concentration, lactant concentration of bacterial biomass per cubic meters of air for every visit of each farm, the 4 farms here. So you can see that from indoor to the 1 kilometer site, we'll have a decrease in concentration of total bacteria. And we can see that from 100 meters to the 1 kilometer sites, the difference in concentration will be significant. And after, you can see here that the downwind and upwind sides, the concentration look high, and they are high, but it's because most of the time those sites are secondary gravel roads, and a lot of cars and tractors are passing by us, so they will rise dust that can contain bacteria, so we're going to collect those dust in our samples, so sometimes it can bias a bit our results. And then if we look at the diversity with the sequencing results, first we can see that we have completely different diversity of bacteria, from the pig farms in pink and from the broiler chicken barns in gray. Then after, we can see as we move away from the farm, so indoor it's the square to the upwind sides, the triangles, even if it's not significantly different, we see that diversity seems to be changing as we go away from the sources, even for the pig farms here in black and a bit in green, and for the broiler chicken barns in red and blue. And again, if it's not significantly different, we can see that the upwind sides tend to cluster together, so they tend to have a different diversity than the indoor and even the fan exhaust and 10-meter sites. Now, if we look at the ARG, this is the sum of the total concentration for every genes that we detected for the 4 farms for each visit, so 3 visits per farm, So you can see for every farm that, again, more we go away from the sources, the concentration will decrease. And that, again, at 100 meters to 1 kilometer side, the differences of concentration of total, the sum of total ARGs will be different from the indoor. And again, we see that we have a background remaining at 1 kilometer size that will vary between 100 to 10,000 ARGs per cubic meters of air. And if you're asking me why there's sometimes 0, it happens at this barn because this day it has rain, and rain will deposit the particles. So at those 2 sites at 100 meters and 1 kilometers upwind, we had rain. We wait a little bit, but the bad was done, so we had less emission at this site. This is why we don't detect an ARGs for the specific visit. So if we separate and we look at different family ARGs, again with the sum, here I showed you for tetracycline, we detected 10 genes. We see the same tendency. As we go away from the farm, we will have a decrease in concentration, again for aminoglycoside genes, and sometimes we don't even detect them, even have like the fan exhaust or even at the 10-meter sites. And it's the same for beta-lactam. I showed you those 3, but for the different family that we tested, it was the same pattern. And now it's another way to present the data. This is for the pig finishing building one. So it's a heat map it's the mean of each visit. And we have every 27 ARGs and the 3 mobile genetic elements that we test. So we really see here the gradient of the concentration that will decrease as we go away from the farm. But we can see, I showed you that tetracycline, the 10 genes detected pretty much have the same pattern, while for aminoglycosid and beta-lactam, it's only one gene that had a weight in the sum that I showed you. Here it's just the same thing, but for the broiler chicken barn number 2, we can see that the pattern of ARGs is different, but again, we will see a different gradient for the different genes, and we will see that for this barn at 100 meters, the concentrations drop rapidly. And I present just one pig and one broiler because the pattern for production type was similar between them. So I said perspective, but it must be like what's next. What's next, so I just present you now, it's like we got a picture of what is happening the day that we sample, but what we want next, it's like if you want a movie, you want to see the movement, the transportation of bioaerosols, which when they are going to be emitted from the barn. So we will need AMR, which is a dispersion model that normally will model the atmospheric pollutant. So we will need meteorological data and terrain data. We will really see the transportation of aerosol coming out the barns, and we will be able to know at which concentration, at which distance the concentration are high, or at which distance the concentration are low, and we will be able to map it around the barn to see what is if there's a village at 5 kilometers from the barn. So it's going to really be helpful to see what's really their behavior of biosol once they're emitted from the barn. So the take-home message is even if the bacterial diversity seems to be similar from the indoor to the outdoor air farms, the profiles of airborne detected ARGs are changing as the distance from the bars the farms are increasing. And like the total bacterial biomass, airborne ARGs concentration generally decrease with distance, but we still have a background concentration remaining at 1 kilometer sites. And if we're not convinced, air can contribute to the dissemination of AMR. Thank you.
Hello, here. Didier Okelo from University Hospital of Besançon in France. Nice talk. Thank you for your data. We can expect from your data that people working in those areas with pigs and poultry have a higher exposure with AMR. Do you have data about the prevalence of the carriage of those people?
Not at this study, but in the lab they did. Some years ago, they tested the nasopharynx of pig workers, and they had a control with like wastewater treated plants workers and naive nasopharynx, and they find different diversity, but for bacteria, and they didn't look that much at ARGs, but they look at some MCR1 colistin gene, and they found in some nasopharynx of pig farmers, but none in the naive one and in the wastewater treatment plants. So, but really they had a different profiles of bacteria in the nasopharynx of pig farmers than the others.
Nicholas Ashbolt, University of South Australia. My question is in relation to the form of your bioaerosols. Were you dealing with just simple water aerosols or actual particulate matter? Because it will make perhaps quite a big difference as to how the body exposure might deal with contact with that, in the sense of inhalation and loss in the lungs, if it's just a water molecule, if you like, holding genes or organisms, versus a bit of dust that will be brought back out and then swallowed.
Yeah, but in farms it's more dusty. Mm-hmm. So, and depending on the relative humidity, the watery content of bioaerosol will change, but really it's really mixed things that is gonna be in air samples of farms. But it's true that their samples that we use, it's gonna catch minimum minimal size, sorry, of particles is 0.5 micrometers. And so we're going to catch everything up that it's going to be sampled. It's really an active sampling. It's like a vacuum.
Just to clarify, so the particle range that you're measuring is from 0.5 up to what size in microns?
There's no upper limit. So, but in some study in our lab, we're gonna measure the particle matters. But for my experiment, we did not.
So it'd be interesting to look at the respiratory size, the less than 10 micron, perhaps. Yeah. Thank you.
Merci. Thank you.
Any other questions?
Hi, thank you for your presentation. Roosmarijn Luiken from the Netherlands, Utrecht University. You saw the background or the downwind and upwind levels more or less the same. Were you able to, or do you know how that relates to levels in less farm dense areas? Because I assume it's quite farm dense, if I understood you correctly.
Exactly. There's our student, Samantha, with purple hair. She's looking at, we call them control sites, so it's more waste sites, and she's working on that. We just received the result from sequencing it, but she's still analyzing our ARG's data for that, so it's data that's gonna be coming, but certainly the goal with my result is to compare with her to really know, to see the signature of the farms compared to like control sites.